morning all. Today we are going to learn about the olfactory system and the olfactory nerve. In the case of lower vertebrates and many mammals such as dogs, the sense of smell is highly developed and they are called macrosmatic. In the case of human beings, the sense of smell is less developed, so it is called as the microsmatic. Introduction to the olfactory system. The olfactory system transmits the sense of smells from the olfactory epithelium of the nasal mucosa to the olfactory cortex of the brain. The following structures are included in the olfactory system: the olfactory epithelium and the olfactory nerves, olfactory bulb, tract and striae, and the olfactory cortical areas. First we are discussing about the olfactory epithelium and the olfactory nerves. The olfactory epithelium is a specialized nasal epithelium which lines the superior one-third of the nasal cavity, which including the roof. It consists of three types of cells: the olfactory receptor cells, supporting cells, and the progenitor or basal cells. The olfactory receptor cells are modified bipolar neurons and they lies vertically between the supporting cells. Their dendrites extends as naked process towards the free surface of the epithelium, where they end by forming bulbous enlargement which is called as the olfactory vesicle. The vesicle process contain cilia. They are called as olfactory hairs, which lies in a thin mucous film covering the epithelial surface. The supporting cells present many microvilli towards the free surface of the epithelium. The mucous film covering the olfactory epithelium, which is secreted by olfactory glands. The receptor cells continuously degenerate and are renewed by the progenitor cells. The ability to regenerate primary sensory neurons is unique to mammalian nervous system. Next we are going to learn the olfactory bulb, tract and striae. The olfactory bulb is a flattened oval mass of grey matter which is lying just above the cribriform plate of ethmoid bone. It consists of an outer cortical zone and inner medullary zone. The cortical zone contains olfactory glomeruli and nerve cells, which forms most of the prominent cellular component of the olfactory bulb. The nerve cells of the olfactory bulb comprises of mitral cells, tufted cells, stellate cells, and periglomerular cells. The medullary zone consists of nerve fibers of olfactory tract. A small group of nerve cells situated at transitional zone between the olfactory bulb and the olfactory tract consist the anterior olfactory nucleus. The olfactory bulb, which continue posteriorly as olfactory tract. When traced posteriorly, the olfactory tract divides into medial and lateral olfactory striae. The point of bifurcation is expanded and forms the olfactory trigon. The medial and the lateral olfactory striae are intermediately related to anterior perforated substance and forms its anteromedial and anterolateral boundary respectively. An intermediate stria is sometime present. It extends from the center of trigon to the anterior perforated substance. where it is sink into the base of olfactory tubercle, which is a small elevation of anterior perforated substance immediately caudal to the olfactory tract. Posterolaterally, the anterior perforated substance is related to the ungus, which posteromedially it is bounded by small bundles of fibers. They are called as diagonal bands of Broca. Next, the olfactory cortical areas. The primary olfactory cortex lies between the anterior perforated substance and the ungus. The entorhinal area comprising ungus and anterior part of the parahippocampal gyrus is often termed secondary olfactory cortex. Next, the neural pathway for sense of olfaction. The axons of the olfactory receptor cells carrying olfactory sensations from olfactory epithelium, the first order neurons, which collect to form small bundles. They are called as the olfactory nerves, which passes through the cribriform plate of ethmoid bone to enter the olfactory bulb, where they terminate into the olfactory glomeruli. The olfactory glomeruli are formed by the axons of receptor cells and dendrites of mitral and tufted cells. Each glomerulus receives inputs from about 26,000 receptor cells and passes this information to the olfactory cortical areas through the mitral and tufted cells. Most of the axons of mitral cells forms the lateral olfactory stria and run to the primary olfactory cortex on the same side, which is located between the anterior perforated substance and the ungus on the infromedial surface of the temporal lobe of brain. Some neurons runs via intermediate olfactory stria to connect with the olfactory tubercle, hence with the limbic system. The axons of tufted cells runs in the medial olfactory stria and crosses the midline in the anterior commissure to form synapses with the granule cells in the opposite olfactory bulb. The entorhinal area, Brodmann's area number 28, which receives few or no tracts or fibers directly but receives fibers profusely from the primary olfactory cortex. Hence, it is sometimes called as secondary olfactory cortex.